We're going to start the paper four, another paper four. You only have 3 days left for the exams. And we're going to do a paper four too. This is in November 2019, which I believe is one of the most toughest papers, which you all feel was very, very difficult. So let's start discussing this. Starting with the first question, which is a simple, straightforward question. It's not a very difficult question. ADH is a hormone that is released into the blood of a mammal when changes occur in the internal environment. State one change in the internal environment of a mammal that leads to the release of ADH. Now, what could cause the release of antidiuretic hormone prevents large volumes of urine. So the blood has to become concentrated. You've sweated a lot, you played basketball, so your blood is now very thick or is concentrated. So decrease in water potential or increase in the concentration of blood. Why? Because we need to now stop the water being passed out in the urine. So we have to prevent the urine being large volume and containing a lot of water. Name the part of the body that releases ADH into the blood. Posterior pituitary gland. That's a fact that we had to know. Then figure 1.1 shows the cell of one of the collecting ducts, and you have the cell ADH from the blood capillary. Here you've got this, and then you've got active phosphorylase, and then you've got the lumen of the collecting duct, and then you've got something known as B, and then you've done something known as A, which you can see it's in the cell membrane. It's a cell of one of the collecting ducts. So what could that be? Now we've got to figure this out, and they're going to ask you a very simple direct question. Name membrane protein A. So that's the receptor for the hormone. And name and cell structure B, which was the vesicle, because cell structure B is what? It's the phospholipid bilayer containing the aquaporins. It's the phospholipid bilayer containing the aquaporins, which are going to come here and going to get attached to the lumen of the collecting duct. So we're going to have all this here, and this is going to result in the water being passed from here, going to enter the cell, and then it's going to go into the blood. So lumen of the collecting duct, they're going to come and attach to the lumen of the collecting duct, where they're going to make the membrane more permeable and allow water to enter from the urine, which was going to be passed out. That water molecule is going to go into the cell and then into the blood. Part was a very direct question. The phosphorylase enzyme stimulates structure B. Describe the response of structure B to this stimulation. Describe the consequences of this response. So direct question: vesicles fuse with cell membrane, aquaporins added to cell surface membrane more permeable to water, water moves from lumen into blood by osmosis, water potential of the blood returns to the set point, and less water lost in the urine. And as we read to question two, which is again a very long story, and that of course gives you all a very, very unhappy feeling or a uneasy feeling. So the stickleback fish, GA, I don't read usually the whole thing. I just say GA. I take the first two words. The stickleback fish, GA, has two distinct forms: the saltwater form and the freshwater form. So first, of course, we read that and we say, okay, so there are two types: saltwater form and the freshwater form. The larger freshwater form is thought to have evolved from the smaller saltwater forms. Okay, so the larger ones we think have evolved over a period of years from the smaller saltwater forms. Both forms have armor plating on each side of the body. The plates are made of bone and contain a high proportion of calcium. Okay, so we've got these some sort of plates which are made of bone and they're found on each side of the body. The ectodisplacin gene. Now, even if you don't want to read this, you can just say the EDA gene codes for a protein involved in the development of armor plates. The EDA gene has two alleles, low armor and high armor. So there are only two possible alleles, low and high armor. So you do this, you write the working and keep on discussing this and having this, reading this when you're in the exam. Three main morphs of armor plating have been described. Complete morph armor plating is found mainly in saltwater form, has many plates from head to tail to cover most of the body, and provides defense against large predatory fish. So the large predatory fish cannot really eat them, so limits the growth. Naturally, if they've got these bones on the outside, it's going to limit the growth of the fish. Then we have the partial morphs. So this is the second variety, found mainly in freshwater form. This was found in saltwater form. This is found in freshwater form. and has a reduced number of plates to cover only part of the body. And then we've got the low armor ones. So the low morph ones are the low morph is found mainly in fresh water. So fresh water, and salt water. So we do a first. I do always do a comparison like that, and has very few undeveloped plates and no body cover. So this was cover most of the body. This was only cover part of the body, and this is no body cover. So you do this comparison, you underline this, and you circle these like I'm doing it, and then you figure out the question. A part of the question says explain why the variation in armor plating in stickleback fish can be described as discontinuous. So immediately you come to your mind is you've got to give me the definition of discontinuous variation. So three distinct phenotypes, one, two, and three. 1, 2, and 3. So three distinct phenotypes, right? So three distinct phenotypes, no intermediates, only one gene, only EDA involved, and not affected by the environment. The environment doesn't affect it. Let's make them in any way different. The environment does, like for instance, we say if there are lots of nitrates in the soil, the plant is going to go taller. Or if it's going to less nitrates, of course, it's going to be then stunted growth. So then we come on to the B part of the question. Now, in 1982, at Loughburg Lake in southern Alaska, the entire freshwater stickleback fish population was accidentally destroyed by humans. So, 1982, something happened, fish population was accidentally destroyed by humans. 1982. 1990, a new population of stickleback fish was found in the lake. Most of these fish had armor plates from head to tail on each side. Suggest why these new stickleback fish have armor plates from head to tail on each side despite living in fresh water. And when something like this happens, something weird happens, I always say think of mutation. So the point is mutation. Saltwater fish had colonized the lake and protection from predators. So even if you had just written mutation, you would have got your 1 mark. It's only a 1 mark question, and the possible answers were because they must have come earlier, that colonized, that come from somewhere, and that now they were all living there, or they were the best protection from predators, so their numbers increased. And as you proceed with the question, from 1990, annual sampling took place in the lake. Each year showed a reduction in the number of individuals with complete morph armor. So numbers started to decrease of the complete morph armor plating, which was from head to tail on each side. This change took place in a relatively short period of time. So 1990, we have 96% of the stickleback fish population had complete morph armor. 1993, 3 years later, then only 39% had complete morph armor plating. And then, of course, it's a very direct question. And you know, whenever there's a direct question on natural selection, there's some points which you need to write in every question without even thinking, and has occurred in this new stickleback fish population. Number one, genetic variation in population. Number two, selective advantage. The ones which have lesser ones were at a selective advantage, so could grow larger, lay more eggs, they survive, reproduce. They survive, reproduce, pass on the advantageous alleles, allele frequency increased. This is called directional selection because one side it's moving towards one different phenotype which is being favored. Low calcium supply maybe, and a reduction in the number of predatory fish. So these are sort of words that you write for everyone. They survive, reproduce, pass on the advantageous alleles, allele frequency is increased, directional selection, selective advantage of fewer armor plates, whatever is now reproducing and the numbers are increasing. So you just talk about those, and the numbers which are decreasing actually they're at a disadvantage, and so their numbers are decreasing. So this was a typical question which required the general answers that we always give for a question on natural selection. Look at question 3, which is again a very direct question. Therapeutic proteins are used to treat diseases. The first purified therapeutic protein used was insulin in 1922. The insulin was extracted from animal pancreases. Since 1982, most insulin has been made by recombinant DNA technology. Explain the advantages of producing human therapeutic proteins such as insulin by recombinant. Yeah, naturally, it is identical to human insulin. Well, when animals we use either pig or we use cow insulin. So identical to human insulin, no allergic reactions. Maybe somebody would be allergic to cow insulin, more rapid response, no chance of developing tolerance. We are naturally, if you keep on giving them pig insulin, they develop tolerance and you would need more of the medicine. No risk of transmitting disease. If you remember from cow and pig insulin, we transmitted diseases like mad cow disease, which was caused by a prion protein. And then, of course, we could genetically engineer bacteria and we could produce them as to meet the supply and demand. So large scale production, then cost of production was much lower because we'd have to slaughter animals and get their pancreas and then of course extract the insulin from that. And then people had no religious objections if somebody was using cow insulin or pig insulin. Muslims would have a problem with pig insulin, Hindus would have a problem with cow insulin. So there were no religious objections to bacteria being used. Question requires a little bit of reading again. Calcitonin is a small protein hormone consisting of 32 amino acids. So they're giving you all this information. You need to register this. One of its function is to inhibit the activity of cells called osteoclasts that break down bone tissue. So small protein inhibits the activity of cells called osteoclasts that break down bone tissue. Calcitonin has been used as a therapeutic protein to treat osteoporosis. In osteoporosis, too much breakdown of bone tissue in older people leads to reduced bone density and a risk of breaking bones. When these people fall down, very easily their head of the femur breaks. So figure 3.1 shows the amino acid sequences of human calcitonin and calcitonin from the salmon fish. So we are comparing the calcitonin, which is the human one, and then salmon, is a fish. So we are comparing the salmon fish. Now position 1, 2, 3, 4, 16 to 32, we have the position, we have the human ones. Now let's look at it very carefully. Now, just for my own information, I started to circle them and I said, okay, let me look at the ones which are the same in human and salmon and the ones which are different. And you see a very interesting phenomena. You see how some are same and some are different, and there is not sort of a distinct pattern that which ones are different and which ones. So we have quite a few which are same, and then we have quite a few which are different. Count these 16 out of 32 are the same. So this is same 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and 16. So 16 out of 32 are the ones which are the same. So you look at this next question. The two amino acid sequences shown in figure 3.1 can be compared. The number of amino acids that occur at the same position in both sequences can be counted and expressed as a percentage of the total number of amino acids present in one sequence. This is called the percentage sequence similarity. Use figure 3.1 to calculate the percentage sequence similarity of human and salmon calcitonin. So show your working. 16 out of 32 into 100 was 50%. Next part was the part 2. Compared to human calcitonin, salmon calcitonin is more biologically active. It remains active in the human body for longer and binds to calcitonin receptors more readily. Bioinformatics was used to identify the more biologically active form of calcitonin to treat osteoporosis. Explain how bioinformatics help to identify salmon calcitonin as a suitable form of calcitonin to treat human osteoporosis. Naturally, what is bioinformatics? It's a database of DNA-based sequences data, and it is for comparison of base sequence data. And naturally, we want to compare calcitonin from this, from this, from this, from different organisms. Similar to calcitonin, what do we want to compare it? We want to compare fish calcitonin, human calcitonin, maybe some other animals calcitonin. So predicting the 3D protein structure, and then the sequences are data pooled from all over the world. So we have massive information on the database, and we can find out which one is the nearest one, which one, of course, then we can predict the protein structure of it, and then we can look at it, and the computer is going to do all this, and then of course we can find out which one would be a favorable one to give as a treatment to people suffering from a specific disease. Then we continue with the C part of the question. Salmon calcitonin to treat osteoporosis made by genetically engineered E. coli bacteria. To produce these bacteria, a plasmid was cut and joined to the new gene to form a recombinant plasmid. The recombinant plasmids were then introduced into the bacterial cells. Name an enzyme that can cut plasmid. Restriction enzyme. Join the Salmon calcitonin gene with the plasmid DNA. So we've cut the plasmid DNA and we put the gene of our interest, and now we need to connect it together, and we need to connect it together using an enzyme here called DNA ligase. So plasmid, cut it, restriction enzyme, add the gene of interest, and then of course glue it together using the DNA ligase. Then identify and explain two properties of plasmids that allow them to be used as a vector in gene cloning. Now this is a very direct question. This has come in many papers. If you remember doing it in one paper, you can do it in any paper. So it's the same mark scheme points which we're going to mention here. Plasmids are small, so can be inserted into cells. So that is why we use plasmids. Then they can replicate independently, so large number of plasmids can be obtained. Then they have restriction sites, so new genes can be added because we're using restriction enzymes. Then they have marker genes, so recombinants can be recognized very easily. And they're circular, so they're stable. So these are the points you can write for any question, in which is a direct question, why plasmids are used as vectors in gene cloning. Then we come to the D part of the question. The gene coding for Salmon calcitonin is introduced into bacteria in a specially designed plasmid called an expression vector. An expression vector must contain a prokaryotic promoter such as the lac promoter. Explain why differences in the control of gene expression in prokaryotes and eukaryotes means that expression vector plasmids must contain a prokaryotic promoter. Now, eukaryotic and prokaryotic promoter sequences are very different. They're not the same. Eukaryotic and prokaryotic, of course, I've used abbreviations. You can't be using these. Eukaryotic and prokaryotic RNA polymerase enzymes are different. You see, the promoter region is the one which is recognized by the RNA polymerase, and only if it recognizes its own promoter, it's going to work there. It's not going to work there. So prokaryotic RNA polymerase binds only to prokaryotic promoter. So this was something in the question. If you didn't even know it, you could have figured this out. So there'll be no transcription. Eukaryotic promoter requires transcription factors not present in prokaryotic cells. Remember, eukaryotes are very, very complex and they have transcription factors. Prokaryotes would not even recognize these and they would not have any transcription factors. So you cannot be using that. So this was very basic biology which we needed to understand. Then we come to question 4, meiosis. Question 4, meiosis has an important role in sexual reproduction. Meiosis occurs during gametogenesis in humans and during formation of pollen grain and embryo sacs in plants. Complete table 4.1 to compare meiosis and mitosis. For both columns, put a tick. If the statement is correct, put a cross if it's wrong, and each box must contain either a tick or a cross. It was simple. Chromosome number is maintained. No, it's not maintained. It's maintained in mitosis. Homologous chromosomes pairs, yes, in meiosis, not in mitosis. Sister chromatids separate, yes, in meiosis, yes, in mitosis. Occurs in prokaryotes, no, nothing. Prokaryotes divide by binary fission. They don't have any meiosis or mitosis. And compare, now whenever we say compare, you have to give me similarities and differences. Compare, if I say differences, then it does differ. But if I say compare, it means similarities and differences. Compare the role of meiosis in gametogenesis to produce sperm cells in humans with the role of meiosis in producing pollen grains in flowering plants. Similarities, both produce 4 cells, both produce haploid cells at the end of the day, both are produce genetically different cells. Then differences, sperm is a gamete, pollen is not a gamete, it's actually a gametophyte. I don't think A levels would know that. Sperm cells have one nucleus, pollen has 2 nuclei. Sperm mitosis, then meiosis, whereas in pollen grain there is meiosis first and then mitosis. Then primary spermatocyte versus the pollen mother cell. So there were some differences and some comparisons. You could have got your 4 marks if you were just smart enough to remember some of the stuff and write it down. To the B part, flower color is important in sexual reproduction of insect pollinated. In the rosy periwinkle, Cathanthus roseus, flower color is controlled by 3 genes, RR, DD, and PP, which interact together to control flower color. It's a drawing of the typical petal, red eye, flower. Well, of course, now we read along. The presence of R allele results in a red pigment in the center of the flower, red eye. So whenever we have RR, it is going to be red eye. Now it was very easy if we could figure this out in the beginning of the question, then of course this whole question was going to be very easy. Now if you look at all these, now what are these? These are all red eye. If it's small r, it's no red eye. So this was simple if you understood the first part. Then of course comes the second part. Then comes to the D and the P. Now of course I've colored them. If it said you had the big D and the big P and the R, so that is the big T, big P, R was here, was the yellow one here. D here, P here, and R here. Now where do we have the big D, the big P, and the big R? We only had it here. So dark pink petals. Then let's look at if you had the smaller ones, R, D. Now we colored them blue. So if you had R, D, and P, now where do you have small r, small d, and small p? Well, that was easy for you to figure out. R, D, P was here, white petals. And then of course we had the white petals. So white petals was the one here when we had the big R, and we had the red eye. So the white petals, because we had the small d and the small p, so we had the white petals here. So you had to really read this again once or twice and figure this out. Where was the red eye? And there were two other things you were talking about, how they were interacting. So I've tried to handle this. I want you all to read this question again and then try to understand it, how I've given this the color coding and how I have figured this out. I hope this is helpful. You can pause the video here and have a look at it again and then read through the question again and do it in your books. Part 2 of the question was again a very direct question. The pigments causing flower color in the rosy periwinkle are formed by a biosynthetic pathway. The RLL mutated to produce the RLL. You know, when is mutation means the base sequence changes and results in another allele. The R allele codes for a non-functional protein. So this is the normal protein which should look like this. Now this one which is made is like this, so it is a non-functional. So explain how the mutations then changes R to small r results in no red pigment being synthesized in the flower of rosy periwinkle. The fact that mutations are of different types, and so of course the mutation that changes R is could be base substitution, frame shift, changes the primary structure, changes the 3D shapes. There could be a stop codon, shortened protein, enzyme unable to bind to substrate, and pathway does not function. Naturally, if that enzyme is not going to work, then how is the pathway going to function? So this is how we were supposed to give this marking scheme. And this is a very typical marking scheme for mostly all mutations which are a result of substitution. So base substitution would have occurred, a frame shift, change in the primary structure, and then change in the 3D shape. Maybe a stop codon has resulted, and so there's going to be a shortened protein. Instead of the full amino acids, only a few will join, and then of course there's going to be a stop. So an enzyme is not non-functional. It said enzyme non-functional, so enzyme is unable to bind to substrate. If it's non-functional, it means it's not going to work. It means the depression where the substrate was going to fit is no longer going to fit. They told you that in the question. It's a non-functional protein, and the pathway which was going to result in that pigment being synthesized will not be made anymore. Another question which challenging, which really checks whether how much biology you know. One mutation that changes the flower color of rosy periwinkle occurs in a region of the DNA that does not code for a polypeptide. Suggest what function is this region of the DNA might perform, is a binding site. It's not actually coding for a gene, but it's a binding site for the, so could be a promoter, an enhancer, maybe a stop codon, maybe even a telomere. So there are regions of the DNA, it says like a region that does not code for a gene. So there are non-coding regions in the DNA. So the gene for protein is somewhere is a different type, and then parts of the chromosome as a promoter or enhancer, or maybe it's a stop codon, maybe it's a telomere. So these were the ones that we were expecting from you when we asked this question. I skipped a few questions. I've come to question 8, but I will be doing the other video and completing the rest of the questions. Question 8 was a simple, straightforward question, not very difficult, but you had to know the chapter on respiration. The passage below outlines the process of oxidative phosphorylation in mitochondria. Complete the passage by using the most appropriate scientific terms. Reduced NAD, you could have said FAD, you could have even said coenzyme. You could have even said coenzyme releases hydrogen atoms to cytochrome carriers. Hydrogen atoms split into protons and electrons, and the electrons are passed from carrier to carrier. Energy from electron transfer is used to pump protons into the intermembrane space. So there's a proton gradient is set up across the cristae, or you could have said in a membrane as well. Then protons diffuse through the channel proteins known as ATP synthase, or you could have said stalked particles, or you could have even said ATP synthetase. The protons combine with electrons and oxygen atoms to form water. So, I mean, this was a very basic 6 out of 6, which most of you should have got, because this really examines the first chapter, which is respiration, and which is all nothing but rote learning. And you did not need to go into any complexities about it. So that completes this video here, and I will continue the questions left in the second video, which follows after this. And thank you very much.